Welcome to Science with Tal. In today's video, we will cover some fundamental mechanisms of neurons, namely signal propagation and signal transmission. According to some estimations, the human brain has about 86 billion neurons, and accordingly the morphology of these neurons can greatly vary from unipolar to multipolar. Furthermore, the signals that neurons carry can serve distinct purposes. For example, when we consider the sensory and motor nerves entering and leaving the spinal cord, the pseudo-unipolar sensory neuron carrying information about touch, and the multipolar motor neuron carrying motor information to contract the muscle, use the same core mechanisms to propagate and transmit the signal. For that reason, to make things simpler for us, we can consider a more general model, one that we will call the Standard Neuron model. By using the figure we have here, I want to first go over a general overview of the anatomical details of the standard neuron, so we can establish the basic terminology that will apply to the rest of the video. Alright, so like any other cell, the standard neuron has a nucleus and all of its typical organelles located in the cell body or soma. One particularity of the neuron lies in its morphology, which is distinguishable by the processes leaving out of the soma. In the schematic, the left-hand side of the soma corresponds to the dendrites or the input. On the right-hand side, one different protrusion from the soma holds a special name, and it is called the axon. The axon starts at the axon initial segment, or axon hillock, and conducts the signal up to the axon terminal, the output. At the output, the standard neuron forms connections with adjacent neurons. The connections are named synapses. The signaling cell is thus referred to the presynaptic neuron, and the receiving cell is referred to the postsynaptic neuron. You can think of presynaptic as in before or pre-synapse, and post as after the synapse. Some neurons may have myelin sheaths to increase the speed of signal propagation, but this is something we will obviously cover later. In all, one can consider the standard neuron as having four distinct functional segments. First, there are the dendrites which receive the signal from external sources. Secondly, there is the soma which integrates the many signals it receives from the dendrites. Thirdly, the axon which conducts the signal to the terminal, and finally the axon terminal which transmits the signal onto the postsynaptic cell. Obviously, there are always exceptions in biology, and some neurons do not follow this general blueprint, but for the most part, neurons function in this manner. I have mentioned a few times that neurons communicate through signals, but what exactly is that signal? Here again, to establish some useful terminology that we will use throughout the video, let me give a very surface-level overview on the signaling mechanisms neurons use. So, the entire signaling system is predicated on the fact that, in contrast to their external environment, neurons are purposefully held in an imbalanced state in terms of ionic concentration, which makes the outside considerably more positive. Due to the charge separation, we say that in such a state, neurons are polarized. Because the ions are charged, we can also consider this charge separation in terms of voltage. Neurons at rest have a voltage in the range of -50 to -90 mV with respect to the outside of their membrane. For simplicity, we will assume that this neuron has a resting value of -65 mV. If, for some external reason, the cell intakes positive ions inside its body, then this will disrupt the resting state, as the voltage difference between the inside of the neuron and the outside will become progressively more positive. As the neuron intakes more and more positive charges, it reaches a certain threshold that opens specific channels that let positive charges flow inside. Consequently, it drastically increases the voltage of the neuron. This event, named the action potential, depolarizes the neuron and gets propagated through the axon up to the axon terminal. At the terminal where synapses occur, the propagated depolarization provided by the action potential allows other channels to open. These channels at the axon terminal allow the entry of calcium, a special ion that allows the release of neurotransmitters in the synaptic cleft. Neurotransmitters are small molecules released by neurons, which go and bind on the receptors of the postsynaptic cell. Generally speaking, neurotransmitters can have two effects which both impact the resting potential of the postsynaptic cell. They can either be excitatory and depolarize the postsynaptic cell, or be inhibitory and hyperpolarize the postsynaptic cell. As you can tell from the graph, a hyperpolarization simply means that the resting potential is further lowered, let's say from -65 to -75 mV. As you can imagine, If enough excitatory signals reach the postsynaptic cell, then the threshold will be reached on the postsynaptic cell and thus cause an action potential to be triggered. From this process, you can see that the signal in question is the action potential that travels from neuron to neuron. Alright, with all of this introductory information now in our toolbox, keep in mind that there are obviously a lot of nuances about the process that I haven't mentioned yet, but no worries, we will get into them later. This overview simply showed the general rule of what happens in the standard neuron. Also, as a side note, since this video aims to explore neurons and their signaling mechanisms, this video will not directly spend a lot of time discussing the organelles you would typically find in eukaryotic cells. Nonetheless, keep in mind again that the usual cellular functions, such as ATP production by the mitochondria, cytoskeletal regulation through actin, microtubules, and intermediate filaments, as well as protein sorting and modification by the Golgi complex, for example, all still occur in the neuron. Anyhow, let's discuss the signaling process in more detail by heading down to the membrane of the neuron. As you might know, the plasma membrane is a hydrophobic barrier that separates two aqueous mediums, the cytoplasm of the cell and the extracellular matrix. As such, the membrane prevents any hydrophilic substance, or, in other words, any water-loving substance, from entering or exiting the cell. One such hydrophilic substance is ions. When dealing with neurons, the most relevant ions we will encounter are calcium, potassium, chloride, and sodium, which all bear their respective charge. Due to the polar nature of water, ions attract water molecules. Indeed, even though water molecules have a neutral charge, the high electronegativity of oxygen creates a net dipole moment that partially shifts the electrons towards the oxygen. This thus creates a partial negative and partial positive charge on the molecule. These partial charges interact with ions in a complementary way, the partial positive with the negative charge and vice versa. In solution, these interactions envelop ions in what we call hydration shells, and these shells are exactly what prevents the ions from crossing the membrane. To cross the membrane, ions go through particular proteins that are embedded in the membrane. Many different classes of these proteins exist, but for our discussion today, we will cover ion transporters and ion channels. When ions flow through ion channels, their motion does not require any energy expenditure because the ions follow their electrochemical gradient, which is a property I will explain shortly. For this reason, movement through these channels is said to be passive. On the other hand, ion transporters move ions against their electrochemical gradient, and this process requires energy, usually in the form of ATP, which makes this an active transporting mechanism. As I've mentioned in the overview, there is an imbalance of ions across the membrane, which results in an imbalance in electric charge. To see where this imbalance comes from, let me first explain what is the electrochemical gradient for an ion. So, regardless of their charge, ions are subjected to two main forces that dictate their movement. The first force being the chemical gradient, and the second being the electric gradient. To see how these forces influence ions, imagine two different scenarios. In the first, there are two equally concentrated solutions of potassium chloride that are separated by a membrane only permeable to potassium. In the second scenario, imagine that the solution on the left-hand side has double the potassium chloride concentration with respect to the right side. Here again, the two solutions are separated by a membrane only permeable to potassium. Obviously, in a physiological depiction, the ions will be disorganized and moving randomly, but for the sake of making it more visually clear, I will group the ions linearly as shown here. Now, what you have to imagine is that the movement of ions is always dictated by how they can equilibrate the system. In terms of the chemical gradient, this means that the ions will always move from a region of high concentration to a region of low concentration in order to balance the amounts on each side. For the electric gradient, this means that ions will always naturally move to the opposing charge in comparison to their own, such that the net charge of the system is as close as possible to neutral or zero. In some way, we can say that for ions, their ideal situation is to be equally as concentrated on either side and also to be electrically neutral as a system. This ideal situation is exactly what is depicted in the first scenario, where there is no imbalance in either charge or concentration, so even though the potassium ions can cross the membrane, there is no net change in charge or concentration. In the second scenario, however, since there are 2 times more potassium chloride on the left side, the chemical gradient will drive the potassium to equilibrate by crossing over to the right. Remember that here, it's only the potassium that crosses because it is the only ion that the membrane is permeable to. By doing so, you will notice that even though now the potassium is equally concentrated on either side, the departure of potassium has left behind many negatively charged chloride ions. This creates a net positive charge on the right and a net negative charge on the left. Since potassium is positively charged, it will be drawn to the left side because it is attracted to the net negative charge created by the free chlorides. In the second scenario, you can see that both the chemical and electrical gradient influence the motion of potassium, and it occurs mainly because there is an uneven distribution in initial concentrations. If potassium continuously flows, at some point it will equilibrate such that the force it feels from its chemical gradient is equal to the force it feels from the electric gradient. As you can see in this example, regardless of how the potassium ions move, it won't be possible for them to be equally concentrated on either side and fully neutralize the charge. Nonetheless, their drive to equilibrate the chemical and electrical gradients is equally as strong, which essentially constitutes a form of equilibrium for potassium. As a result of the difference in charges between the inside and the outside of the cell, we can establish the membrane potential, which corresponds to the voltage across the membrane, or in other words, the difference in voltage between the extracellular matrix and the cytoplasm. In our example here with potassium, we can also establish its equilibrium potential, which is the membrane potential at which the drives from the chemical and the electrical gradients are equal for the ion. The equilibrium potential for any ion can be mathematically computed with the Nernst equation. In this equation, the equilibrium potential for an ion is equal to the gas constant multiplied by the temperature in Kelvin divided by the valence of the electron, which is essentially its charge, multiplied by the Faraday constant, and all of this multiplied by the natural logarithm of the outside concentration of the ion divided by its inside concentration. When certain conditions are assumed, for example 37 degrees Celsius, the equation can be simplified to 26.72 millivolts times the natural log of the concentration over the valence. We can further simplify the equation by converting the natural log to a base 10 log by multiplying by about 2.3. The conversion will make the calculations a bit more convenient and more intuitive later on. In all, this leaves us with this equation, which we can now use to compute the equilibrium potential at 37 degrees Celsius for each ion that contributes to the membrane potential. To do so, we need the concentrations in and out of the cell for each ion and their valence. With data taken from this textbook, we can compute the equilibrium potential for each ion. You will notice that, let's say for sodium, the concentration inside varies from 5 to 15. Hence, the equilibrium potential also varies. You will also notice that I have added anions to the table. In this case, anions are negatively charged amino acids and proteins that influence the net charge of the system, or in other words, the membrane potential, but since they cannot move directly through the membrane, we will not consider their equilibrium potential. As you can see from the equilibrium potential column, the values greatly change depending on the ion. Sodium, for example, would be perfectly at equilibrium if the membrane potential was between positive 89 and 60 millivolts, whereas potassium would be at electrochemical equilibrium if the membrane potential is at negative 88 millivolts. As we will see time and time again in this video, this contrast in equilibrium potential between potassium and sodium is exactly what fuels the propagation of the signal. Now, an important detail we have not discussed yet is how exactly do the ions end up distributed the way they are, and most importantly, how is this distribution maintained across the membrane? As you will see throughout this video, the most important ions when it comes to signaling and conducting the action potential are potassium and sodium. For that reason, let's first cover what mechanisms the cell uses to separate these two ions. The creation and the maintenance of the sodium and potassium gradient is performed by the sodium-potassium transporter, also known as the sodium-potassium pump. During a cycle of this protein, it exports 3 sodium ions for every 2 potassium ions it imports. From what we've mentioned about transporters, remember that moving ions against their concentration gradient requires energy. In this case, the energy to move potassium and sodium comes from the hydrolysis of ATP. You can notice that one consequence along with the creation of concentration gradients is the creation of a net positive charge on the extracellular side due to the extra positive charge exported in the sodium. Hence, it will be good to keep in mind that this protein also contributes to the membrane potential. Now, for the other ions, there are other transporters that are responsible for their gradients. In terms of the calcium gradient, there is a calcium pump that utilizes ATP hydrolysis to pump 2 calcium ions against their concentration gradient, as well as importing 2 protons. Furthermore, calcium can be transported in the extracellular matrix through the sodium-calcium antiporter. Remember that to pump an ion against its electrochemical gradient, it requires energy. Antiporters are a class of ion transporters that generate sufficient energy by coupling the transport of another ion that goes down its electrochemical gradient. In this case, sodium, which we know is more concentrated outside, will have no problem flowing inside through the protein. In contrast to antiporters, which export and import ions, you can also find symporters, which either export or import ions in the same direction. For example, the potassium chloride symporter uses the movement of potassium to send chloride in the extracellular space. Here again, the protein uses the energy released by potassium flowing down its electrochemical gradient to fuel the process and extrude chloride. In summary, ion transporters, regardless of how they get their energy, are very important proteins that allow the generation and the maintenance of electrochemical gradients. Now, back to the membrane, let's quickly summarize what we've established. Thanks to the ion transporters, each ion is now differently concentrated across the membrane. As a result, the ionic separation causes each ion to build an electrochemical gradient of its own that now greatly influences whether the ion wants to leave or enter the cell. Mathematically speaking, we were able to obtain a value of the equilibrium potential for each of our ions thanks to the Nernst equation. Now, remember previously that we have established the membrane potential, which was the difference in voltage between the extracellular matrix and the cytoplasm. Unfortunately for us, even though the equilibrium potential is a good indicator to know for each ion, it does not allow us to attribute a value to the membrane potential. This is mainly due to the fact that the Nernst equation does not take into account the ionic interactions that occur at the membrane. Now, what do I mean by these interactions and how will we be able to quantify them to get a value for the membrane potential? Recall in the second scenario, where I demonstrated the electrochemical gradient, that the membrane was only permeable to potassium. So, since potassium was the only ion that was able to move across the membrane potential in this system was solely dependent on the equilibrium potential of potassium. Indeed, given that it is the only ion that can move to equilibrate the forces that it feels from the electric and chemical gradient, when the potassium will be at its equilibrium potential, then the membrane potential will also have no other choice but to be at the equilibrium value of potassium. In this scenario, however, if the chloride was able to move across the membrane, then its motion would have imposed its own electrochemical gradient on the membrane potential. In other words, the movement of chloride would have shuffled the ions in a different way and thus led to a different membrane potential. Another scenario we can think of is what would happen if the membrane was, yes, permeable to both ions, but considerably more permeable to potassium. In this case, the equilibrium potential of potassium would influence the resting membrane potential considerably more than chloride, simply because potassium ions can flow at a greater rate. So, back to my question, which was, what do I mean by the ionic interactions? As you can see from my explanation, the main factor of ionic interaction is the level of relative permeability each ion has with respect to the other ions. In other words, the ions that are able to flow the most will have the greatest influence on the value of the membrane potential. For that reason, To get an accurate value of the membrane potential, we need to turn to the Goldman equation, which factors the relative permeabilities of ions. The Goldman equation is very similar in shape to the Nernst equation, and it makes sense since we're still trying to figure out a voltage for our system. The first difference with the Nernst equation you will notice is the insertion of the permeability factor. The permeability factor, which has units of velocity, measures the rate of movement from the ion driven by a local concentration gradient. In other words, it is simply a measure from 0 to 1 on how great the ionic flux of the specific ion is. Another difference you will notice is the sum symbol, which simply indicates that this product of permeability times the concentration has to be present for each ion inside the logarithm. Now, back to our system, you can see in the right corner that I've written the long form of the Goldman equation and that I've simplified it in the same way we did with the Nernst equation by assuming a temperature of 37 degrees Celsius and changing the natural logarithm to a base 10 logarithm. You will notice that the concentrations for chloride are inverted. This is because the valence of chloride is -1, so with the properties of logarithms, we can invert the numerator and denominator to get rid of the negative. From experimental data, scientists have established that the permeability ratios for each ion at rest are the following. Potassium is valued at 1 because it is the reference the scientists took to arrive at the other values. You will notice that calcium is at 0, and that is just because it doesn't really affect the membrane potential at rest, so it is usually neglected from the Goldman equation. Since all the values we have for either the concentrations and the permeabilities are at rest, the membrane potential we get from the equation is what we refer to the resting membrane potential. And from our data, its value is -72 millivolts. As a quick note, regardless of if you use 5 or 15 for the intracellular sodium concentration, the resting membrane potential will only change by a few decimal because of sodium's very low permeability at rest, so we can essentially neglect the range of sodium. Now that we have the membrane potential, we can establish a new quantity known as the driving force. This quantity is obtained by taking the difference from the resting membrane potential and the equilibrium potential of the ion. The driving force is essentially a measure of how much the ion wants to move in or out of the membrane. As you can see, potassium at rest has a very low driving force compared to sodium, since the membrane potential is predominantly set by the permeability of sodium. Keep the driving force in mind because this property will be very important for us in our future discussions. In summary, the resting membrane potential is governed by two factors. First, the considerably higher permeability of potassium relative to the other ions sets the resting membrane potential very closely to the equilibrium potential of potassium. Secondly, the sodium-potassium pump contributes a bit to the charge separation by pumping one extra positive charge in the extracellular matrix. As a result, it contributes to setting the resting membrane potential a few millivolts below. The most important function of the pump, however, is to maintain the concentration gradients for each ion. Without its constant action, the gradients would dissipate and the signaling mechanisms of the cell would all be damaged. Now, before we move on to the passive membrane properties of the cell, I want to take a quick minute to introduce ion channels and their properties because it will be relevant in upcoming sections. When it comes to ion channels, they have two important properties that must be established. First, what ions go through them? Is it sodium, potassium, chloride? And this property is usually referred to as their selectivity. And secondly, what type of gating maintains the channel closed? For example, is it a voltage, a tension, or a chemical that will open the channel? When it comes to the selectivity of the ion channel, the current model rests on two properties that are both located in the pore of the channel, namely the size of the pore and specific binding sites in the channel. To exemplify the logic of the mechanism, let's see how different channels filter between sodium and potassium. Recall that ions are covered by a shell of water molecules. It turns out that the smaller an ion is, here sodium relative to potassium, it creates a bigger hydration shell because the charge is more concentrated and thus creates a stronger electric field to attract more water. Potassium channels can utilize the size property and have a specific pore size such that the potassium can cross, but sodium cannot, along with a specific binding site preferable to potassium. In sodium channels, it is thought that the pore has a binding site that favors the binding of sodium considerably more, such that only sodium can cross the channel. In both cases, the pore size and the binding sites play a role in the selectivity. Now, for the gating properties of the channels, channels generally have two conformations, either closed or open. The transition between the closed state and the open state is what is referred to as gating. In relation to what we have discussed in the section about selectivity, when a channel opens, it generally allows to widen the pore size and position the binding sites such that the ions can efficiently cross the membrane. Three major mechanisms exist to gate ion channels in neurons. First, ligand-gated channels can be opened by the binding of a chemical also known as a ligand. One very important example for us are neurotransmitters, which are a specific category of ligands that we will thoroughly cover later. Secondly, voltage-gated channels can be opened only when a specific membrane potential allows them to. For example, voltage-gated sodium channels will only open when the membrane potential is at a certain threshold, let's say negative 40 millivolts and above, but when it is below like it is at rest, the channels will stay closed. These channels have a few more intricacies that are important for us, but we will cover them in due time when we will discuss the action potential where that information is most relevant. Finally, the opening of tension-gated channels is regulated by mechanical force or tension. Such channels are prevalent in the auditory system, in the stereocilia of the cochlea, and piezo channels in the somatosensory system. These channels often have some form of physical linker that is attached to the channel, either intra or extracellularly, which allows them to mechanically change the conformation of the channel. Now, back to the Goldman equation. As I have discussed in the overview, the generation of a signal in the neuron begins with the entry of positive charges in the membrane, or in other words, a depolarization. We now understand that the positive charges that enter the cell are sodium, and they are able to enter because their permeability has increased. The entry of these charges causes a disturbance on the resting membrane potential, which we just calculated with the Goldman equation. Unfortunately, one downside of the Goldman equation is that it cannot tell us how the disturbance caused by the depolarization will impact the membrane potential across time and distance. For us to understand how such disturbances impact the resting potential, it can be best understood if we use an electrical circuit model to map our neuron, which is often referred to as the equivalent circuit model. Alright, to make the circuit, we must first establish how the biological components in the neuron relate to the basic structures that we can find in electric circuits. As we have covered, due to its hydrophobic nature, the plasma membrane creates a charge separation between the two conductive mediums by acting as an insulating material. Hence, we can consider the membrane as a capacitor, which is represented by this symbol in electrical circuits. The capacitance C, generated by the membrane, is equal to the stored charge divided by the voltage across the capacitor. The units for capacitance are farads, which are expressed as an F. Because the membrane contains ion channels that let charges move, the membrane is considered a leaky capacitor. The ion channels in this case are a source of conductance because they allow charges to flow. Recall that resistance is the inverse of conductance, so we could technically think of the channels in terms of resistance. In this case, the resistance could be thought of as the fact that the ions cannot freely cross the membrane. However, I will rather keep the conductance because it is more intuitive to think of how channels allow the current to pass rather than how much they impede it. Nonetheless, I will use the symbols of resistors to show the conductors, so make sure you don't get confused with this element. The units for conductance are Siemens and are represented with S. The missing component that we need to establish now is what is the battery of our system, or in other words, what is the source of voltage. To exemplify this property, let's consider a small circuit for one potassium channel. In this simple circuit, you will recognize in orange the potassium channel shown as the resistor symbol. This conductor has a conductance value of lowercase g that is associated with the potassium channel. Okay, Now let's consider a first scenario where there is no potassium gradient across the membrane. In this scenario, the only force that acts on the potassium to push it across the membrane is the membrane potential created by the other ions. As such, the force, or in terms of electrical circuit, the battery, is the resting membrane potential, Vm. From Ohm's law, the current across a conductor is equal to the product of the conductance and the voltage. Hence, in the first scenario, the current across the potassium channel is equal to the conductance of the channel multiplied by the resting membrane potential. Now, imagine a second scenario where we only consider the chemical gradient from potassium. From what we went over in our discussion on the electrochemical gradient, recall that in this scenario, the motion of potassium will be influenced by two forces. First, the chemical gradient that drives the potassium to exit, and secondly, the electrical gradient that forces the positive charge to equilibrate the negative charges it leaves behind. For that reason, the equilibrium potential acts as a battery to supply energy to drive the potassium movement. Now, let me mention that by convention, we consider positive current as the movement of positive charges out of the cell. In this scenario, since we consider the chemical gradient, we can expect the potassium to flow out of the membrane. Hence, the outward movement of potassium corresponds to a positive value. Here again, we can use Ohm's law to describe the current through the channel, but to make sure the signs align with each other, the negative sign on the right side must be present because the equilibrium potential of potassium is negative, and we expect a positive current. Finally, let's consider the scenario where the concentration gradient and the membrane potential both act as forces on the potassium. In this scenario, the net current through the channel will be equal to the sum of the currents found in the scenario 1 and 2. By factoring the conductance, we arrive at this expression where the current is equal to the product of the conductance and the difference between the membrane potential and the equilibrium potential of potassium. If you remember from our previous discussion, this difference is what we know as the driving force, which we vaguely define as how much the ion wants to enter the cell. But to see what this equation means, let's consider it in more detail by plotting it on a graph. As you might notice, this equation has a linear shape with the current being the dependent variable and the membrane potential being the independent variable. Let's first imagine that the membrane potential is equal to the equilibrium potential of potassium. From what we know from previous discussions, the equilibrium potential for potassium when the concentrations are set as they are right now is in the range of -70 to -90 millivolts. Regardless of what the actual values are, if the two potentials are equal to one another, you can see that the current will be equal to 0. This makes sense because it means that the electric and chemical forces acting on potassium are perfectly balanced and no net movement is occurring. Now, let's imagine that the membrane potential is higher, let's say 30 millivolts. Then we would expect a current value to be positive. Recall that a positive value of current means that the potassium is leaving out of the channel to the extracellular side. On the other hand, if the membrane potential is lower than the equilibrium potential, then the current will be negative and indicate that the potassium is entering the cell. This graph is usually referred to as an IV plot, and it allows to illustrate in which direction current flows depending on the voltage. This is a graph that we will see again and again throughout this video. For terminology's sake, you might sometimes see the equilibrium potential be named as the reversal potential because it is the value beyond which the current direction reverses. Back to this figure, we now have all the basic analogs to properly compare the neuron to a circuit. I just want to mention that the voltage is in units of volts and that the current is in units of amperes. Now let's build an electric circuit that represents the neuron. As we've seen, the important ions that contribute to the resting membrane potential are potassium, sodium, and chloride. Also, we have seen that their channels all have different selectivities and permeabilities. Thus, we must consider each type of channel as separate conductors. As we did with potassium, each conductor is connected to its battery, or in other words, their equilibrium potential. In the potassium example we just did, we only considered the conductance of one channel. To take into account the entire population of channels, we can multiply the number of channels by the individual conductance to get the total conductance, here noted as capital G. To complete the circuit, we can add a few more important elements. First, we can add the active pumping of the sodium-potassium pump, which is essentially a current generator that keeps the batteries charged, and we can also add the membrane capacitance, which is denoted as Cm. Thirdly, we can add the membrane potential to the circuit, but it is more often shown in this discrete way, where these two hollow dots correspond to the short circuit symbols with the extracellular matrix on one end and the cytoplasm on the other. The final thing to add is the direction of the currents. Now, let's use our circuit and try to find an alternative equation for the resting membrane potential. To simplify the derivation, we can neglect the sodium-potassium pump because its influence on the membrane potential is very small. Also, since we are trying to get a value for the resting membrane potential, we can neglect the membrane capacitance as well. Let's start from the fact that since we are trying to compute the membrane potential, we know that there will be no net flux to the system because it is in equilibrium. In other words, we can translate the statement as there will be no net flux in current, which means that the sum of all currents going through the channel will be equal to 0. Secondly, we have already established in our previous discussion that the current going through each channel is given by the modified Ohm's law, where the voltage is replaced by the driving force. From there, we can solve for the membrane potential by replacing each current with their definition in step 2. Then, we can apply a bit of algebra and finally isolate Vm. The final equation we get is very reminiscent of the Goldman equation, except here we deal with conductances instead of permeabilities, but overall they both give a value for the membrane potential. Remember that the purpose of modeling the neuron as an electric circuit was for us to be able to quantify the effects of depolarizations on the membrane potential. Beyond the little introduction I made about neurotransmitters at the beginning, we do not know quite yet the mechanisms responsible for depolarizations. One thing for sure though is that these depolarizations do happen and they are what initiate signals. With our new model of the electric circuit, these depolarizations essentially constitute an injection of current into our circuit. As we've seen, such injection of current caused by, let's say, neurotransmitters must reach a threshold in membrane potential to activate the action potential and consequently propagate the signal. The action potential is often referred to as an active response because of its all-or-nothing nature, whereas the current injections that first build up to the action potentials are qualified as passive responses because of their graded nature. Indeed, passive responses vary in size and are not sustained like action potentials. That is to say, they decay over time if they do not reach the threshold. What I'm trying to emphasize here is that the generation, let alone the propagation of the signal, is entirely dependent on the voltage of the cell. So if you don't reach the threshold, you don't get any action potentials. And the only way that the voltage can change is through these current injections which produce depolarizations. For that reason, the build-up to the action potential in the passive phase is just as important as the active phase in order to properly understand the signaling mechanisms of neurons. With this being said, I want to point out that the circuit that we have built for the moment does not clearly discriminate between what happens during passive or active conditions. Indeed, the passive graded state, when we don't consider ligand-gated channels that open from interactions with other neurons, mostly operates through what we call leak channels, which includes a wide variety of potassium, chloride, and sodium channels that are open at rest according to the relative permeabilities we have established. On the other hand, the active all-or-nothing state operates through voltage-gated potassium and sodium channels that are only open at a specific threshold. Hence, you can see how this circuit can get a bit confusing because it does not discern between active and passive channels. To correct it, I will use this circuit instead, where the purple channel represents the currents coming from all the leak channels, and the two other channels are the active voltage-gated channels. As we will see later, the conductances in the active channels change as a function of time and voltage, which is illustrated by these arrows that cross the resistor symbols. As a sort of preemptive note, In the section that we are about to begin, we will focus on understanding how, in the passive phase, the membrane potential changes in response to a current injection. To do so, we will use our circuit model to arrive at equations that describe the behavior of the membrane potential as a function of both time and space. Along the way, we will discuss three important passive membrane properties. To generally describe them, passive membrane properties are intrinsic properties of the neuron that influence how the membrane potential of the neuron will change in response to a current injection as a function of time and distance. We have already seen two of the three properties. The first property is the membrane capacitance. The second is the membrane resistance or conductance, depending on how you see it. And the third property is the axial resistance. For the moment, I know that they do not mean much beyond what we have discussed, but as we go through the equations, these properties will become much clearer. Alright, so to understand these properties, let's begin small by greatly simplifying the circuit we have made. For the moment, let's not even think about the ion channels and only consider the capacitance of the cell. Also, let's assume that the capacitor is discharged. That is to say, it does not store any energy. If we inject positive charges in the system, the positive charges will create an imbalance of charges on the capacitor. Because of charge repulsion, the addition of a positive charge on one side will effectively kick out another positive charge on the other side. Due to the created movement on the opposite side, this creates a capacitive current. We will denote the injected current as capital II and the capacitive current as capital IC. Furthermore, the charge imbalance, which we can note as delta Q, creates an electric field across the membrane which produces a potential difference of delta V. Delta Q and delta V can be linked together through the membrane capacitance in the equation delta Q equals Cm times delta V. Remember that this is just another way of writing the equation I first showed you when I introduced the membrane as a capacitor. The goal for us now is to see if we can find an equation that will model the change in membrane potential as a function of time. This will allow us to see how the membrane potential evolves over time with a current injection. First, we can establish the definition of the current, which is the net movement of charge per unit time. In calculus terms, this corresponds to the derivative of charge with respect to time. Secondly, we know from Kirchhoff's current law that the sum of all currents at a node is zero. Because of our conventions, the injected current will be negative since there are positive charges going inside the cell. As such, the capacitive current is equal to the injected current. We can replace the capacitive current by its definition dq over dt. Then, we can replace dq by its definition. We know that in our case, dVm is the membrane potential, and since the membrane capacitance is a constant, we can take it out of the derivative. Now, we are left with a differential equation that we can easily solve, yielding this equation. To make things simple, we will assume that the current injected is held constant, and thus the change in charge is simply described by the constant current we inject times the amount of time we have injected it for. To get a better feeling of the equation we just derived, let's see it graphically on the plot. You will notice that the equation we have is linear, so we can expect a linear behavior from our graph. Alright, on the first plot, let's show how the injected current evolves over time, and on the second plot, let's show how the membrane potential evolves over time. At first, let's assume that there is no injected current and that the membrane potential is simply given by whatever resting value it already has. Then, at some initial time t1, we inject current up to a value I0. As we can see in the equation, the current injection will impact the membrane potential in a linear way up to a point where we turn off the injected current at some point t2. Because we are considering a scenario with no ion channels, the charges that have entered the cell cannot leave, so the membrane potential will maintain its value as long as there is no other current injection. But we know, however, that the cell is not simply made of a plasma membrane, and that it also has a lot of channels on its surface, which essentially makes the membrane a leaky capacitor. Hence, we can add the leak channels as resistors to our model. Usually we use the conductance value, but for reasons that you will see later, I will use the resistance right now. In this scenario, RL corresponds to the leak resistance, or in other words, the total resistance of the cell coming from all of the leak channels in the membrane. We will also assume that the current going through this resistor is positive and goes out of the membrane. We can make this assumption because we know that at rest most of the current is coming from potassium, which has a tendency to leave the cell. The current from this resistor is IL. Interestingly, you can note that now our model of the neuron is essentially an RC circuit. As we did for the simple case with no resistor, let's find an equation that will show us how the membrane potential changes over time. Here again, we can start from the two basic equations we've established previously. Likewise, in this parallel RC circuit, we can establish that the sum of the currents in a node is equal to 0. Thus, with our convention, we can formulate that the injected current is equal to the capacitive current plus the current coming from the resistor. As we previously did, we can substitute the currents with more thorough definitions. For the resistive current, we can use Ohm's law. From there, we can multiply both sides by RL to get this expression. In this differential equation, we can find the steady-state solution, which occurs when the rate of change in membrane potential equals 0. With dV over dt being 0, it leaves us with Vm being equal to the injected current times the resistance of the membrane. Here, we can establish a new quantity called Vmax, also known as V infinity, which corresponds to the maximal voltage the cell can get at a steady state. With this new definition of Vmax, we can replace the expression on the left by the definition. Next, we can establish a new quantity named the time constant, which is represented by the Greek letter tau. This constant is essentially the product of the membrane resistance and the membrane capacitance and has units of time. We will see shortly how this constant applies to our system. Anyhow, let's substitute the constant in the expression, and now we have a differential equation that we can solve. You can go through the steps on your own, but essentially we arrive at this expression. Here again, to understand how this equation operates, let's plot its evolution over time along with the injected current. You can notice that the equation we have is exponential and not linear anymore. This equation is a bit messy to read, but one way you can think about it is dividing it in 3 sections. In blue, we have the membrane potential of the system as a function of time. And in green, we have the maximal voltage that the membrane potential can attain. The tricky part is the exponential bit in pink. You can think of the pink segment as a factor that tells you how much the membrane potential in blue approaches Vmax. Indeed, the pink factor on its own is an increasing function that gives a value between 0 and 1. Thus, as t increases, so does the fraction of Vmax. To make it more intuitive, let's see how this applies with an injection of current in our system. Here again, we'll assume that the injection of current is constant over a short interval t1 and t2. On the plot of Vm, we can indicate in dashed lines what would Vmax look like as a function of time. As we've established, Vmax is equal to the injected current times the leak resistance. Since the injected current and the leak resistance are both a constant, then so is Vmax. Therefore, as a function of time, Vmax will stay a constant and will have the same shape as the current. Now, for the actual membrane potential, at time t1, we can consider it as t equals 0 with respect to the current injection. By plugging 0 in the membrane potential equation, the pink term becomes 0 and so does the membrane potential. Now, as time goes by, the pink term progressively increases and a larger and larger portion of Vmax is attained up until Vm pretty much equals Vmax at t2. As Vm ascends to Vmax, remember that the time constant has units of seconds, and at some point the time will be equal to the time constant. When that happens, the value produced by the pink term is around 0.63. In other words, At the value of T equals tau, we say that the membrane potential is 63% of the maximal voltage. When the current is turned off at T2, the membrane potential progressively returns to the baseline state according to a similar equation. The equation is a bit different because it describes an exponential decay instead of an exponential growth, but it still has the same essential components. Now, to understand more in depth why the voltage exponentially grows and exponentially decays, let's consider how the currents from the capacitor and the resistor operate in our model. When the current is injected in the cell, it first goes to the capacitor to charge it because the capacitor imposes the least amount of resistance to the current. But as it gets charged, the current going through the capacitor progressively diminishes because the capacitor is getting full and thus the current feels more resistance. Simultaneously, the current going through the resistor progressively increases since it now becomes the path with least resistance. When the current is turned off, the capacitor discharges a current that is compensated by the resistor. Since the voltage is mainly set by the current of the resistor, the shape of the voltage as a function of time will be the same as how the resistive current evolves for both the growth and decay parts. With this in mind, we can also analyze the system through the lens of the time constant. What happens when it is high or low and so on. First, let's establish a reference value in blue. Consider now a lower time constant because either the resistance or the capacitance is lower. In the increasing portion of the membrane potential, a lower value of tau means that as time goes by, a smaller fraction of time will be divided and thus the pink term will increase faster relative to the reference tau. As a result, the membrane potential will arrive faster at Vmax and discharge faster as well. With a higher tau, the opposite relation is true, which is that the potential will take longer to charge, but also longer to discharge. Hence, you can see that by using a value of the time constant, we can compare neurons to see how the signals are maintained in them. You probably remember that there is another component to the channels, which is the driving force coming from the electrochemical gradient that is associated with each ion. When we include the driving force and do the math following the same principles as in previous scenarios, you can see that the model ends up being the same. The difference simply lies in the expression of Vmax, which now is offset by the Nernst potential of the leak channel. Hence, even with the battery added, it is still the same relation. Now, I have to tell you that the model we have just built to predict how the membrane potential changes with a current injection only works when we consider the neuron as a simple spherical shell that has a uniform voltage across its volume. Fortunately for us, this model can very well approximate what happens with current injections in a soma, but for compartments such as the dendrites and the axon, this model does not hold anymore. And we need something else to describe the behavior of the membrane potential. For example, when we consider the dendrites, the voltage following a current injection drastically differs as a function of position. As a result, voltages across the entire tree of dendrites differ at a given time, whereas the voltage at the soma stays relatively uniform. The main reason why the voltages differ in terms of position is because the charges leak out to the exterior while they are traveling in the dendrite. But we will come back to that aspect shortly. As I've pointed out before, dendrites are often the actual sites of current injection via synapses or other sensory mechanisms, so to properly understand how voltages are impacted in dendrites, we need to create a new electric circuit model that will accommodate their difference. The structure of dendrites can be very complex, but to approximate their shape in 3 dimensions, we can model them as cylinders. When we consider the inside of the cylinder is still part of the neuron, so you can imagine it as having a phospholipid bilayer shell and the inside filled with cytoplasm. We will assume that the cylinders have a constant radius A, and we will attribute a value L to the length of the cylinder. Note that the length of the cylinder is in the axial axis. To make a better approximation, we can segment the cylinder into small units of length delta x. But before we dive into the circuit, I want to take a minute to explain the terminology of the electrical components such that it doesn't get too confusing later. First, let's establish a few geometric equations. What will first be relevant for us is the area of the cross-section of the cylinder, which is simply the area of a circle, so pi times the radius square. What is also relevant for us is the area of the surface of the cylinder, and we don't need to take into account the ends of the cylinder. Hence, the surface area is simply 2 pi times the radius times the length of the cylinder. Now, regarding the actual electrical parameters, remember that our cylinder is made out of a plasma membrane, so here again, our cylinder will generate some form of capacitance by separating positive red charges from negative blue charges. If we consider the total capacitance of the cylinder, noted CT, we can determine the membrane capacitance per unit length, noted lowercase cm, by dividing the total capacitance by the length. We can also determine the membrane capacitance per unit area, also known as the specific capacitance, by dividing the capacitance per unit length by the circumference. This quantity is noted as uppercase cm. One simple relation we can already establish with our model is that if we decrease the radius of the cylinder, we decrease the size of our model dendrite, and thereby we reduce the amount of charges that the membrane can store. Hence, the capacitance is proportional to the size of the cylinder. In addition to the capacitance, remember that the membrane has channels embedded in it that allows ions to flow in and out of the cell. As such, we can establish the same quantities for the total resistance, resistance per unit length, and resistance per unit area. Here, however, it is a bit tricky because it seems like the areas are not at the correct places. This is because resistance is inversely proportional to the area, and thus increasing the area reduces the amount of resistance because with a larger membrane there are more channels that ions can take to flow out of the cell. To get the opposite relation, you can consider the conductance, which is proportional to the area. So as you increase the surface, you increase the conductance. One final property we now have to consider is the resistance due to the cytoplasm as the charges move inside the cylinder. This resistance is named the axial resistance and is noted capital RA. Similar to what happens in an electrical wire, we can define this internal resistance as the product of the resistivity of the cytoplasm times the length divided by the area of a cross-section of a cylinder. Here again, we can find the axial resistance per unit length by dividing the total resistance by the length, which leaves us with this expression. Just like the membrane resistance, the axial resistance is inversely proportional to the size of the cylinder. Indeed, if the cylinder is smaller, the area for the charges to flow becomes more constricted and thus makes the flow of charges harder. To have a more intuitive picture of this relation, you can imagine how the flow of water in a pipe would be influenced if the diameter of the pipe changes. In both cases, increasing the size reduces the internal resistance. Now, this is a lot of small equations, and we are not going to use them all. Here are the relevant properties and equations we will need to keep in mind. You will notice right here that these are the 3 passive membrane properties that I have introduced a section ago. Alright, let's get started on our electric circuit model, which will take place at the membrane. The model we are about to begin describing is from cable theory. Here again, we can consider the extracellular matrix and the cytoplasm as 2 conductors. Contrarily to the equivalent circuit model, we need to indicate on the intracellular side the axial resistance. Now, there is a resistance that you can technically attribute to the extracellular side, and it is relevant when the extracellular space is restricted, such as in nerve fiber bundles, but we will just assume that for simplicity, our neuron is isolated and the extracellular space is pretty vast. With this assumption, the extracellular axial resistance becomes negligible. Since we are still dealing with the membrane, we need to indicate the membrane resistance and membrane capacitance, which are connected in parallel to our conductors. Remember that we have segmented our membrane in short segments of length delta x. Hence, each segment of length delta x will have its own little circuit. Also, since each component is defined as per length, we need to multiply them by the length of the slice to get an accurate value, except for the membrane resistance, which is divided because of its inverse proportionality. Now, what is missing for us to complete the circuit is to indicate the currents and the voltages of our system. To understand the currents and the voltages in this system, let's think conceptually of what happens in the cylinder when we inject a current into one region of length delta x. Assuming we are injecting positive charges inside and the resting state is negatively charged, the site where the current is injected will have the highest voltage with respect to the outside with, let's say, a value V0. Now, as the charges begin to spread out, they will have three choices for where they can go. First, the charges can continue flowing through the cytoplasm. We will name this current capital I. Another option for the charges is to leak out of the membrane through ion channels, or finally they can be stored by the membrane as capacitance, which both constitute a form of membrane current noted as Im. As charges continue moving in the cytoplasm, they are faced with that same decision, and they ultimately leak out of the membrane through the channels, which progressively decays the voltage back to a resting value. Both the current and voltage diminish as a function of time and distance. Back to our electric circuit, to make it less convoluted, let's just analyze one junction. One junction will be sufficient to derive the cable equation, but keep in mind that this pattern is repeated many times. So, assuming that the left circuit is at position x, we can indicate the voltage across the circuit as V of x and t. In the right circuit, the voltage will be V of x plus delta x and t. Likewise, we can indicate the currents in the same fashion. Here again, we will adjust the currents that are per unit length by multiplying them with the length. The final piece before we start discussing the cable equation is to indicate the injected current, which again is per unit length. Note that the current does not come from within the cell, but having the arrow like this will make the derivation more intuitive with regards to Kirchhoff's law. Alright, we now have every component to derive the cable equation. Let's start by analyzing the voltages through the perspective of Ohm's law. The difference in voltage between the point x and x plus delta x is equal to the axial resistance times the current going through it. If we divide both sides by delta x, we arrive at a very special equation. Indeed, notice that on the right side, if we take the limit as delta x goes to infinity, we almost have the definition of the partial derivative of the voltage with respect to x. If we add a negative sign, we get the same expression which we can replace in our equation. Thus, we arrive at the relation that the rate of change in voltage with respect to position is equal to the negative product of the axial resistance per unit length and the current going in between. This bit of information will be very useful for us, so I will keep it close. Now, we can also analyze the currents of this system with Kirchhoff's law, which stipulates that the sum of the currents at one node equals 0. Because it is positive charges moving, remember that our convention says that positive charges moving out of the cell is positive current. And in the horizontal plane, we will assume that the right is positive. Next, we can rearrange the similar terms that are multiplied by delta x together. Here again, we can divide both sides by delta x, which gives us an expression very similar to the partial derivative of the current with respect to position if we take the limit of delta x to 0. This, as a result, gives us that the membrane current leaking out minus the injected current is equal to the negative partial derivative of our current with respect to position. Remember that the negative sign here is to adjust the definition of the derivative. The expression we have previously found comes into play. If we take the partial derivative of both sides, we can arrive at the fact that the partial derivative of the current with respect to position is equal to minus 1 over the axial resistance times the second partial derivative of voltage with respect to x. Consequently, we can replace the partial derivative of current to get this new expression. Remember from our discussion on the equivalent circuit model that the current going through this parallel system of a capacitor and a resistor is simply equal to the capacitive and resistive current, which are expressions we have already found. Since the voltage is now a function of time in position, we need to use the partial derivative and not the simple derivative. Anyhow, we can replace the membrane current by this expression, which leaves us with this. The final step in our derivation is to multiply both sides by the resistance, which gives us this final equation. To simplify it a bit, we can make two constants. First, the time constant, which we have already discussed at length in the previous section, and the second constant is the space constant, denoted by lambda. The space constant is defined as the root of the membrane resistance divided by the axial resistance. This constant has units of length. Another quick note, since E is a constant and can be essentially treated as an offset, you will often see this equation without the E in order to make it simpler. In all, the equation that governs how the membrane potential changes as a function of time and space in our model cylinder is this very complicated second-order partial differential equation. Nonetheless, to get a good feeling of the equation and the space constant, we can consider a simpler scenario where we disregard time, otherwise known as the steady-state scenario. In this scenario, we will have to make a few assumptions. First, we assume that the length of our cylinder is infinite. Secondly, we assume that we inject current directly in the middle of our cylinder, such that there is only a value of current, I0, at the origin, and everywhere else other than x equals 0 has a current value of 0. Thirdly, as it says in its name, the steady-state scenario disregards time, and assumes a scenario where the rate of change in voltage as a function of time is equal to 0. In our context, this implies that the capacitor is fully charged. By imposing the condition that del V del T is equal to 0, our partial differential equation now simply becomes a differential equation as it only takes position in consideration. The solution for this equation when we disregard the current is the following. The membrane potential as a function of distance is equal to an initial voltage times an exponential factor that varies according to the position. Like in our discussion about the time constant, we can think of this equation as having three compartments. The blue segment of this equation is the membrane potential as a function of distance, the green is the maximal voltage that is measured at the origin, and the pink term is the decaying factor. The decaying factor in pink will decrease for all x, so as the position gets further from the origin, then so does the voltage. Let's visualize this equation by plotting it on a graph. At x equals 0, or in other words, the site of current injection, the pink term becomes 1, and thus the voltage has a maximal value of V0. As you get further from the site of injection in either direction, the value of the membrane potential is less and less impacted by the current impulse because of the leak current. When you are at a position equal to lambda, you are at a position where the membrane potential has decayed to 1 over E of its value, which is about 37%. Thus, the space constant gives a measure of how far the disturbance in Vm extends from the site of current injection. To see its impact more graphically, let's consider the blue curve as our reference lambda. If we were to consider a smaller lambda, you can see that the curve becomes smaller and means that the voltage has decayed faster than the reference value. On the other hand, a higher value of lambda shows that the signal decays slower from the site of injection, which means that the membrane potential can be impacted further. One interesting observation about the space constant is what factors mostly influence its value. When we look at the equation of the constant, remember that the membrane resistance per unit length is simply the membrane resistance per unit area divided by the circumference, and the axial resistance is the resistivity divided by the area of a circle. After substitution and simplification, you can see that the constant is directly proportional to the root of the radius. Thus, as the dendrite or axon gets bigger, then so does the space constant, and thus the signal is better maintained in terms of position. With this being said, we have now covered the three passive membrane properties: the membrane capacitance, the membrane resistance, and the axial resistance, as well as the two constants, the time constant tau and the space constant lambda. In summary, these properties affect how the magnitude of the membrane potential changes as a function of both time and distance. One final note to consider about the two constants is that the time constant, which is given by the product of the capacitance and the membrane resistance, is solely a property of the membrane, whereas, as we've seen, the space constant is dependent on the radius, and thus it is a geometric property of the neuron. Anyway, it is good to keep these properties and constants in mind, as we will come back later to them when we will go over the mechanisms to increase the speed of conduction of the action potential and how the soma integrates many synaptic inputs. With the passive properties now covered, we are ready to go over the mechanisms behind the action potential. To properly understand the action potential, we must begin our discussion by describing how the first recordings of this event even came about. For a long time, the action potential was a very hard phenomenon to measure because as soon as the membrane potential hits a certain threshold, the action potential fires and there is no way to maintain a specific voltage to measure the conductances of the channels involved. The major breakthrough in understanding the action potential down to the electrophysiological level came from experiments made by Alan Hodgkin and Andrew Huxley. These two scientists used the voltage clamp technique to successfully record the currents and conductances that make up the action potential. To appreciate the results and measurements, let me explain how the voltage clamp apparatus works. To do so, let's take a chunk of the axon and place it in a dish. Contrary to most organisms, squids have very big nerve cells that can have diameters up to 1,000 times larger than mammalian axons. Hence, the large diameter axon of the squid allows the experimenters to insert electrodes and other electrical equipment without rupturing the cell. For that reason, let's assume right now that we are currently working with the axon of a squid. So, in the first portion of the apparatus, one electrode is placed in the axon and the other electrode is placed in the baiting solution. The two electrodes are then connected to an amplifier which measures the membrane potential. The voltage measured in the amplifier is then connected to another amplifier that compares it to a desired voltage. The desired voltage is often referred to as the command voltage and it is generated from a signal generator. The second amplifier then sends a compensatory current to the axon through two electrodes such that the voltage of the axon is always kept at the command potential. And whenever it is different, current is either sent or withdrawn. The current that goes into the system can then be measured with an ampere meter. To better understand the technique, imagine that we set the command voltage at a higher value than the membrane potential. In this case, to match the command voltage, the membrane potential must get more positive, which implies that the feedback amplifier will inject current into the neuron. In the other scenario, where we set the command voltage lower than the membrane potential, then the membrane potential will have to get more negative to match the command voltage, which is accomplished by withdrawing current from the system. Hence, the apparatus continuously provides a negative feedback loop that keeps the membrane potential at the same value as the command voltage. Now, before we examine more thoroughly the different results obtained from the voltage clamp experiment, I want to give you a reminder that the axon is composed out of three main types of channels. There are voltage-gated potassium and sodium channels, as well as leak channels that are always open at rest. These three categories of channels will be of interest for us in this section. Moreover, recall that the membrane current flowing out of the cell is composed of a capacitive and a resistive current, which constitute the opposite of the injected current. One positive aspect of the voltage clamp technique is that it essentially removes the capacitive current. Indeed, as we've defined it, the capacitive current is equal to the capacitance of the membrane times the rate of change of the membrane potential with respect to time. But if we hold the membrane potential constant at a value equal to the command voltage, then the capacitive current equals zero. Thus, when the voltage is quote-unquote clamped, the injected current directly equals the resistive current from the three types of channels at the membrane. Now that this is established, let's consider more concrete examples. For the two first examples, let's plot the membrane potential and the membrane current as a function of time. On the plot of the membrane potential, let's assume that the resting membrane potential is at negative 60 millivolts. While we haven't touched the command voltage yet, the membrane is at rest, so there is no recorded current. In our first trial, let's set the command potential at negative 70 millivolts to slightly hyperpolarize the membrane. Before we see the results, remember that since the command voltage is below the membrane potential, the voltage clamp will withdraw positive current, or in other words, send negative current into the system. For the split second that the resting membrane potential adapts to the command voltage, the capacitive current flows and causes this high negative peak. Then, while the axon is clamped, the current that will flow is only from the leak channels until we turn off the system and the membrane readapts again with the capacitive current. In the second trial, let's set the command potential at negative 50 mV to slightly depolarize the membrane. As expected here, we get the opposite relation, with the currents flowing in opposite direction and with the leak channels still being the only ones responsible for the resistive current. Recall that our convention for current says that positive current corresponds to positive charges leaving the cell, which in essence corresponds to a hyperpolarization. Accordingly, negative current corresponds to positive charges entering the cell and depolarizing the cell. In voltage clamp recordings, positive currents are often referred to as outward currents and negative currents as inward currents. Consequently, in the first example, we say that the leak channels have an inward current, while in the second example, the channels have an outward current. The logic behind this, which ties everything together, is that let's say in the first scenario, the apparatus is trying to make the cell more negative, but the cell wants to be at rest. So to fight the injected hyperpolarizing current, the current going out of the leak channels will be depolarizing, or in other words, inwards. In the second example, it is the opposite logic. The cell becomes more positive, but wants to be more negative. Thus, it will produce an outward current to hyperpolarize the cell. The take-home message from these two examples is to exemplify the convention of voltage clamp recordings that an inward current represents a depolarization and an outward current represents a hyperpolarization. These definitions are very important because I will keep using them in the next few sections. Alright, now for the third trial, let's induce a very big depolarization by setting the command potential at 0 mV. You can see that now the results are very different from what happened before. Nonetheless, here again, as the membrane potential equilibrates, there is a brief capacitive current that is recorded. Then, it is followed by a very brief inward current, and then an outward current that persists until it reaches a certain plateau. Through this curve, you can also imagine that there is a certain constant current coming from the leak channels that still flows along. As you can see, the considerably different response between trial 2 and 3 shows that there is a certain voltage dependence. From these results, we can establish that the ionic permeabilities change drastically as a function of voltage and time, but it doesn't tell, or at least indicate to us very clearly, what ions are involved in the process. To get more intuition about the process, one can perform the voltage clamp experiment at different values of command voltage. With each trial, you can notice that the inward current gets smaller and smaller as the command potential increases up to the point at 60 mV where it isn't even present anymore. To understand why this happens, recall that the equilibrium potential of sodium is about 60 mV. Hence, when the axon is clamped at 60 mV, there is no net flux of sodium, which indicates that the inward current is most likely controlled by sodium influx. Another line of evidence of sodium's involvement in the process comes from another experiment where the sodium is removed from the extracellular solution. The removal of sodium from the outside solution now makes the equilibrium potential for sodium very negative. Indeed, when the voltage is clamped, there is no early inward current, but rather an early outward current, which further proves that sodium is responsible for the inward current under physiological conditions. This experiment also shows that even without outside sodium, there is still a prolonged outward current, which is caused by another ion. It turns out that this outward current is caused by potassium, which can be proven in multiple ways such as radioactive labeling. But one very compelling proof comes from studies using pharmacological products. Indeed, when one performs the voltage clamp with Tetrodotoxin, which is a toxin that blocks voltage-gated sodium channels, you can see in the results that only the delayed outward current is recorded. You will also notice that we are not considering the capacitive current anymore because it doesn't give any relevant insights on how the action potential works. Alright, in another trial, when one uses tetraethylammonium, a drug that inhibits the voltage-gated potassium channels, the recordings show only an early inward current, which we know sodium is responsible for. Thus, we can deduce from these results that potassium is indeed responsible for the outward current. Therefore, when we look back at the original recording, the first bit of current is the transient sodium inward current, which is followed by a delayed outward potassium current. Now that we have a better grasp of which ions are responsible for the action potential, the next step for us is to understand how the conductances of these ions change over time and voltage. As a refresher of what conductances are, recall our equivalent circuit model, which we use to model passive membrane properties. In this circuit, the total membrane current IM is given by the sum of all the currents, which include the capacitive current from the membrane and the three currents coming from the three channel types. We have also established in previous sections that the current that flows through these channels is equal to the conductance times the driving force. From these equations, we can thus get an expression for the conductance of each ion, which is simply the current divided by the driving force. To illustrate how the conductances evolve over time and voltage, let's consider some voltage clamp recordings. So, for a range of membrane potentials, let's simply consider the shapes of the current and the conductances without even thinking of any numbers to see what relations we can establish purely from the curves. Let's begin with sodium. In terms of current, we know that at negative 50 millivolts, there will be no current going through the sodium voltage-gated channels, since the threshold will not have been reached yet, and thus the channels will still be closed. Accordingly, the conductance through the channel will also be zero. Then, as we pass the threshold to, let's say, negative 20 millivolts, we get the typical inward sodium current. Here, because there is a current flowing, we also record a value of conductance. At 0 millivolts, the current and the conductance both increase again, but interestingly, at positive 20 millivolts, the current decreases while the conductance increases again. To understand what happens here, let's use the equation of current as a reference. So, as the membrane potential increases, the conductance increases because it is a property of the channels, and the channels themselves are increasingly voltage dependent. Remember, however, that the equilibrium potential of sodium is about 60 millivolts, and thus, as the membrane potential increases, the driving force decreases because Vm and the equilibrium potential of sodium are approaching one another. Indeed, when we consider the plots at 60 millivolts, you can see that there is no net current, even though the conductance is very high, and that is caused because the driving force is zero. On the other hand, when we consider potassium, because its equilibrium potential is very negative, the current and the conductance keep increasing as the membrane potential increases. To better appreciate these results, we can overlap the potassium curves with the sodium ones. From these results, there are two similarities and two differences between the behaviors of sodium and potassium that we can establish. In terms of similarities, notice that the two channels require a threshold and membrane potential to open. Indeed, at some values, like here at negative 50 millivolts, there is no conductance, which implies that the channels are voltage gated. Moreover, you will notice that for both channels, the conductance is proportional to the step in depolarization. When we consider the peak conductance as a function of membrane potential, the relation for both ions is sigmoidal in nature. At a very negative membrane potential, the channels are closed, and no ions cross, whereas at very positive potentials, the conductance plateaus at its highest. We will later see how to obtain this relation. In terms of differences, you can see that the sodium channels open more rapidly, and after a short time they inactivate. On the other hand, the opening of potassium channels is more delayed relative to sodium and remains sustained. As a result, the time courses of these two ion channels are very different. To understand why voltage-gated potassium and sodium channels have different voltage and time dependencies, we need to consider the individual structure of the channels and how they function by themselves. For the moment, the currents that we have considered are currents that correspond to the summed input of thousands of channels. To record currents coming out of a single channel, scientists use the patch clamp technique. In this technique, a small micropipette, which contains an aqueous solution of varying concentrations, is pressed against the membrane of the neuron. After applying a bit of suction to the back of the pipette, it allows to create a very tight electrical seal on the edges, such that there is very little ionic leaks in and out of the pipette. A metal electrode that is connected to an amplifier is inserted inside the pipette. The other terminal of the amplifier is connected to the command voltage. The output of the amplifier makes a loop through a feedback resistor, and the current is measured at the output as well. As you can see, the patch clamp is essentially a voltage clamp setup, but instead of clamping the entire cell, the experimenter clamps a single patch of cell containing only a few channels. One important upside with the patch clamp is that the technique has various configurations that allows the experimenter to vary the experimental conditions more freely. The current setup that we have right here is usually referred to as the cell-attached patch clamp, and it allows to directly measure the current from individual channels. From the cell-attached patch clamp, if the pipette is retracted while the seal is still established, it will remove the attached bit of membrane from the cell. This configuration is known as the inside-out configuration and has the benefit for the experimenter of having control over the internal medium. Again, from the cell-attached recording, one can increase the suction to a point that will break the membrane. In this configuration, also known as the whole cell patch clamp, the pipette is continuous with the interior of the cell, which allows to record measurements of the potential inside the entire cell. From the whole cell patch clamp, retracting the pipette with the seal still established will, here again, break the membrane, but interestingly enough, it will reform on itself to create this outside-out configuration. The outside-out configuration is very important when the experimenters want to have control on the extracellular medium. This is usually a good technique to measure currents going through individual ligand-gated channels. In summary, there are 4 different configurations of the patch clamp method that all have their particular advantages that the experimenter can utilize. To see how this technique works, let's first do an example of an outside-out recording on the leak potassium channel to see what results we can draw from it. In our system, imagine that the concentration of potassium inside the pipette is 140 millimolar and the outside is 5 millimolar. With these values, which closely resemble what happens physiologically, we can compute the equilibrium potential for potassium through the Nernst equation, which gives us a value of negative 88 millivolts. Now, let's use the patch clamp technique to clamp the system at various voltages. For each command voltage value, the microscopic current recordings in picoamperes as a function of time will look a bit like this. As a side note, here microscopic simply refers to the fact that the current comes from one channel. As you can see, microscopic currents are quite different from the usual continuous currents we are used to see. Indeed, microscopic currents turn on and off, and that is caused by the fact that channels essentially behave like random switches. When the channel is closed, there is no current passing through, and as soon as the channels open, a somewhat constant value of current flows through. The number of times the channel opens and for what duration is entirely probabilistic. So on the individual scale, ion channels behave somewhat very randomly. Now, to understand the direction and the magnitude of the currents, we need to go back to a relation we have previously established. Remember that leak channels are simple resistors, and the current flowing to them is equal to a constant conductance times the driving force. As such, the results here directly showcase this relation. For example, in the third trial, the membrane potential is clamped at the equilibrium potential, and thus there is no net flux of potassium. Between trials 1 and 2, the increase in current simply comes from the fact that the driving force for potassium has increased. And in the fourth trial, the inward current happens because the driving force has switched signs. Using the physiological concentrations in and out for sodium, we can perform the same analysis with the leak sodium channels. Here again, the x-intercept corresponds to the equilibrium potential. Now, what is more interesting to us, however, is to see if the patch clamp method can give us a bit of insight as to why the macroscopic current from voltage-gated potassium channels is so different from the current coming from voltage-gated sodium channels. It turns out that when one performs multiple trials of the patch clamp experiment on both voltage-gated channels, the individual currents coming from the channels have the same general differences. The potassium current is delayed and sustained, whereas the sodium current opens first and rapidly inactivates. This entails that the difference we are looking for lies in the molecular structures of each voltage-gated channel. Another important point here is that with voltage-gated channels, it now becomes tricky to make them an IV curve because, as we saw, the conductance changes as a function of time and voltage. Nevertheless, we can arrive at a good model that explains both the time and voltage dependence of conductance if we first consider the molecular structure of voltage-gated channels. So all voltage-gated cation channels generally have this repeated motif of 6 transmembrane alpha helices, termed S1 to S6, and an intermediary P region located between S5 and S6. In this motif, the S4 segment is thought to act as the voltage sensor because it contains a pattern of positively charged amino acids that are only present in voltage-gated channels. The P region forms the selectivity filter, which is basically the property of the channel that dictates which ions cross. To get more information on the mechanism behind the selectivity filter, remember that we have discussed how it works in our discussion about the general properties of the ion channels. The voltage-gated channels for sodium, calcium, and potassium have 4 of these motifs. One difference between these channels, however, is that voltage-gated potassium channels are tetramers, meaning that the motifs aren't connected to each other, whereas voltage-gated sodium and calcium channels are linked together. One particularity with sodium channels is that they have an important motif named the inactivation motif. We will later see how this motif affects the channel. Alright, now that we have a general sense of the structures behind these channels, let's examine how we can model the voltage and time dependence in the current equation. Let's first start with the voltage-gated potassium channels because they are a bit easier to understand in contrast to sodium channels. The 4 large cylinders here correspond to the 4 motifs. Recall that ion channels generally have 2 states, either closed, like it is right now, or open, which requires a conformational change in the channel structure. When the channel is open, ions can flow through the selective pore. From the results of the voltage clamp experiments, we know that voltage-gated channels are closed at rest, for example when the membrane potential is at -70 mV, and that they are open upon depolarization when the membrane potential is at, let's say, 10 mV. The mechanism that opens and closes the channel comes from the voltage sensors. At rest, the positive charges in the voltage sensor feel the electric field generated by the negative membrane potential and thus are drawn to the intracellular side of the membrane. When the sensor is in that position, it induces a closed conformation. As the cell begins to depolarize, the positive charges in the voltage sensors now feel repulsion from the cytoplasm and start moving towards the extracellular matrix. The change in conformation is believed to be responsible for twisting the alpha helices such that the P region of all subunits is in the correct conformation, which opens the channel. One very important thing to note here is that the voltage sensors do not all move together at the same time. Remember from the patch clamp recordings that the opening of ion channels is stochastic, which means that even under a depolarization, the channels can stay closed because the opening relies on the probability. Hence, when we think of the conductance, which relies on the channels being open, we can describe it as the product of the probability of the channels being open times the maximal open conductance, here represented with a capital G with a bar. Since the maximal conductance is a constant, you will notice that now the voltage and time dependence have shifted on the probability. When we consider the voltage sensors that make up the channel, the same probabilistic nature applies. For each subunit or motif, we can associate a gating variable n, which represents the probability that the subunit will be open, or in other words, that the voltage sensor is in the correct conformation. The probability of the channel being open requires that all 4 voltage sensors are in the correct conformation. And because the voltage-gated channel is a tetramer, we can assume that each subunit acts independently from each other. Thus, the probability that the channel is open equals n to the power 4. By substituting this expression in the current equation, we can obtain this new expression of the current. This is a good step forward, but obviously that did not solve our issue because it simply shifted the dependencies of the conductance onto the gating variable. Nonetheless, the mathematical model becomes much simpler to obtain when we only consider the gating variable. Alright, let's start by considering the time dependence for one gating variable only. When we consider the probability n of a channel being open, we can also consider that the probability to be closed is 1 minus n. The change between the open and closed state can be modeled as a simple rate equation with 2 transition rates. The first rate, alpha n, describes the transition from closed to open, and the second rate, beta n, describes the transition from open to closed. Both rates have units of per second. The time dependence here arises from the fact that the conformational change, from closed to open or vice versa, involves thermal activation. When Hodgkin and Huxley developed this model, the two scientists assumed that the rate of change of the gating variable with respect to time could be described as this first-order differential equation. At first glance, this equation seems to come out of nowhere, so let's take a moment to think conceptually about what it means. First, we can simply think of dn, which is the change in the gating variable. By itself, the gating variable n corresponds to the probability associated with the open state. Hence, we can broadly think of the change in that probability as the difference between the number of closed subunits that open and the number of open subunits that close. Therefore, for a given change of time dt, the number of closed subunits that open is simply the rate at which the closed gate becomes open times the proportion of closed subunits. Inversely, the number of open subunits that close for a given change of time dt is equal to the rate at which the open gate closes times the proportion of open subunits. With this, we get the differential equation from Hodgkin and Huxley. By rearranging the equation and dividing by alpha n plus beta n, we can get this expression in which we can obtain 2 important values. First, we can consider the steady-state solution, which happens when the derivative of n with respect to time is equal to 0. This leaves us with 0 being equal to alpha n divided by alpha n plus beta n minus n. Here, we can establish a new quantity called n infinity, which corresponds to the maximal probability of opening the gate at a steady state. Secondly, we can establish a time constant tau n, which corresponds to the inverse of the sum between alpha n and beta n. This term has units of seconds. With these 2 new variables, we can greatly simplify the differential equation to this expression. This form of the equation is a bit more intuitive to understand. Basically, this equation says that at any fixed voltage, the rate at which n is reaching the steady-state value depends on the time constant. If the time constant is large, then it will take longer to reach the steady-state value as opposed to a smaller time constant, which will make the process quicker. When we assume that the voltage is at a fixed value, the solution to this differential equation corresponds to this. Before we analyze this equation in more detail, remember that the transition rates alpha n and beta n greatly depend on voltage. Consequently, n infinity as well as the time constant are also voltage dependent. For that reason, let's briefly cover the voltage dependence, and then we will come back to this equation to properly analyze what all of this means. In relation to the time dependence, it is a bit more tricky to obtain the voltage dependence because when Hodgkin and Huxley were building their model, they went with a data fitting approach. meaning that they had a bunch of results for alpha n and beta n, and they made up their own curve that would fit their results instead of directly deriving the equation. The equations that they found for alpha n and beta n are the following. With these last two equations, we now have every tool to analyze the potassium current. You will notice that some signs might be inverted with respect to the original paper. This is simply to fit the voltage conventions that are used today. Alright, so to put everything together, let's plot the transition rates and n infinity as a function of voltage, as well as n as a function of time for a given depolarization. Let's start with the transition rate curves. According to Hodgkin and Huxley's equations, the shape of alpha n and beta n looks something like this. As the membrane potential increases, alpha n exponentially grows and beta n exponentially decays. We know from a measurement standpoint that as we depolarize the cell, the voltage-gated potassium channels start to progressively open, so it makes sense that the rate at which they open increases and the rate at which they close decreases. When we look at the curve of n infinity as a function of membrane potential, we see the sigmoidal relationship that we have previously seen with the potassium conductance. To see the relevance of this curve, let me also show you the time-dependent results. So, for a voltage clamp-held membrane potential, n infinity will be a constant since it only depends on voltage. According to the solution we have, n will relax exponentially as a function of time up to the value of n infinity evaluated at Vm equals 0. Because we know that conductance is proportional to n raised to the power 4, it is more fitting to show how n to the power 4 behaves as a function of time. As you can see, n to the 4 has a sigmoidal shape, just like the shape of conductance as a function of membrane potential. In relation to the data from the voltage clamp experiments, we can now understand that the sigmoidal behavior of the conductance directly comes from the behavior of n, which is explained by all of these equations. This is a good step forward, but to understand more thoroughly the behaviors of these voltage-gated channels and ultimately the action potential, we must now consider the behavior of the voltage-gated sodium channel. Like we did for the potassium channels, let me first start by explaining the molecular details, and then we will jump into the mathematical model. Alright, so just like voltage-gated potassium channels, sodium channels can either be closed or open. Voltage-gated sodium channels, however, have a third functional state in which they inactivate. The inactivation comes from the inactivation motif, which bears positive charges and also feels some repulsion. That repulsion will cause the motif to clog the channel and inactivate it, thereby preventing ionic flow. You will notice that in this model, 3 out of the 4 subunits contribute to opening the channel, and the 4th contributes to the inactivation. Now, let's consider the individual subunits of the channel to build up the mathematical model. Just like for potassium, we can rewrite the conductance in terms of a variable probability times a maximal sodium conductance. In terms of single subunit probabilities, we can attribute the letters m and h to the open state. Hence, the closed state corresponds to 1 minus m, and the inactivated state corresponds to 1 minus h. Since 3 subunits contribute to the open state from the closed state, and 1 to the open state from the inactivated state, our probability assuming independence becomes m cubed times h. As a side note, remember that the structure of a voltage-gated sodium channel is not like the voltage-gated potassium channel. The subunits are not separated into a tetramer, and thus, the mechanisms behind the channel do not technically function upon independence, but assuming independence is good enough for our model. Anyhow, with this equation, we can get a new expression for the sodium current. Also, with the same assumption that these states function on the basis of a reaction rate, we can establish for each gating variable an alpha and a beta transition rate. Here again, the alpha rate goes from closed or inactivated to open, and the beta rate goes from open to inactivated or closed. These transition rates now allow us to derive the same equations we have made for the voltage-gated potassium channels, starting with the rate of change of the respective gating variable, which allows us to find the time-dependent solution. Here again, the voltage-dependent relations of alpha and beta for m and h were all found using the data fit approach. All these equations now allow us to analyze the time and voltage dependence of the voltage-gated sodium channel. To keep things simple right now, let's analyze the equations for M and H separately, and then we will compare what they mean relative to each other to get a good portrait of the channel. Starting with M, it is the easiest to understand because it behaves exactly like the gating variable for potassium. Again, we know that upon depolarization, these channels start opening, so it is logical that the rate at which the channels open increases and the rate at which the channel closes decreases as a function of voltage. Accordingly, M infinity follows the same sigmoidal shape as N infinity. We will later see how these curves compare to potassium because there are a few key differences. In terms of time, M also exponentially increases up to M infinity under a clamped voltage, and to get a better description of the entire channel, one can consider M cubed as a function of time. But we know that as a function of time and voltage, the sodium current does not just steadily increase, it shuts off rapidly after activation. This is where H becomes important. Let's start with the voltage dependence of H. By plotting the equations derived from experimental data, you can see that the curves for alpha H and beta H are inverted relative to the transition rates of M and N. Consequently, the graphs for H infinity and H as a function of time are also inverted. The way to interpret this is that at rest, the H gate is open and then closes as voltage increases due to the inactivation motif clogging the pore of the channel. On their own, seeing these graphs gives good insight, but what I want to do right now is compare everything together to get a clear picture of what's going on. So to tie everything together, let's recall what we are aiming to explain. From the voltage and patch clamp recordings, we have seen that the sodium current, given by this expression, yields a fast inward current that rapidly inactivates, and the recordings on the potassium current yields a delayed outward current that persists as long as the depolarization is maintained. To set the baseline, let's briefly summarize the variables from the potassium channel. In terms of gating variables, the potassium channel has only one, which is N. The transition rates alpha N and beta N have this relation in terms of voltage where, as the membrane potential got more positive, the rate alpha to open the channel increased and the rate beta to close the channel decreased. When we plot the steady solution as a function of voltage, the curve for N infinity is sigmoidal. At a fixed voltage, the time relation caused by the thermal fluctuations behind the conformational changes result in the actual value of N to exponentially relax to the value of N infinity as time advances. Now, for the sodium channel, as I've said previously, when we consider M, it behaves just like N. The main difference, however, is that relative to the rates of potassium, alpha N and beta M grow and decay much faster. As a result, the sigmoidal shape of M infinity is more steep than the curve of N infinity, and thus explains why sodium channels open before potassium channels. In terms of time, M reaches M infinity much faster and thus also illustrates how sodium channels open first. When it comes to H, you will notice that when we look at H infinity, it is very high at negative potentials, which means that the inactivation gate is open at rest. For the entire sodium channel to be open, both M and H need to be open. The issue is that as voltage increases, H decreases and M increases. For that reason, the maximal probability that the channel will be open will occur at a voltage where the two maximal values meet, which will be somewhere around -20 millivolts. As a function of time, the same relation happens. The H gate starts high and then progressively decays, whereas M progressively grows. If we consider the shape of M cubed times H, you will notice that in the rising phase, while H is high and M rises, their product also rises. But after some point, when H starts to considerably decrease relative to M, the value goes back to zero even though M is high. Notice that the shape of M cubed times H is what gives this complex shape to the conductance of sodium. Now, for us, because we know what the molecular structures of the channels are, The inactivation process of sodium and the sustained current from potassium seem trivial, but you have to imagine that Hodgkin and Huxley did not know anything about these structures, yet they managed to accurately model the currents from sodium and potassium because their results fitted when they used n to the 4 and m cubed times h. So even though they did not know what the structures of voltage-gated channels were, they were still able to accurately describe the physiological process behind the action potential, which we can now do as well given the information we have on the channels. So here, let's imagine that we are looking at a very small section of the axon and paying attention to the behavior of the voltage-gated sodium and potassium channels. To keep track of what's going on, let's have a small plot that illustrates the membrane potential and the conductances for each ion as a function of time for this small subsection of the axon. Alright, assuming that for whatever external reason the neuron gets excited and the membrane potential increases, it will have the effects of gradually opening the voltage-gated channels through the voltage-gated sensors. Because of the intrinsic properties of the voltage-gated sodium channel, it will open faster to its maximal capacity relative to the voltage-gated potassium channel, and thus let sodium enter first in the cell. As the conductance of the sodium channel increases, the entry of positively charged sodium further depolarizes the cell and pushes the membrane potential up to the equilibrium potential for sodium. This depolarization phase caused by sodium, more commonly referred to as the rising phase, is then stopped by three factors. First, as we've discussed, the driving force on sodium becomes much less because the membrane potential approaches the equilibrium potential for sodium very closely, and hence the sodium has less of a drive to enter the cell. Secondly, the sodium channels progressively inactivate, which causes the conductance of the sodium channels to gradually decrease. Finally, the delayed voltage-gated potassium channels now open at maximal conductance and start repolarizing the cell towards the equilibrium potential of potassium. As the membrane potential goes back to the resting value, the potassium channel closes and its conductance decreases as well. Note that potassium channels do not open at the peak of the potential, they open earlier around at the negative 20 millivolts range. For terminology's sake, the repolarization is known as the falling phase, and the peak of the action potential is known as the overshoot. You will notice that the hyperpolarization by potassium pushes the potential below the resting membrane potential. This peak is referred to as the undershoot. The negative potential in the undershoot is then corrected by the sodium-potassium pump that reestablishes the gradient that sets the resting membrane potential. One key factor of the action potential that I haven't mentioned is the threshold in voltage that triggers it. The threshold is a very interesting property because a fraction of a millivolt can be the difference between generating an action potential or not. When the cell is initially depolarized, the inward sodium current is balanced by an increase in two outward currents, from potassium and the leak channels because their driving force increases as the membrane potential gets more positive. Hence, below threshold, the net current is outward, but at the threshold value, which is always higher than the resting membrane potential, the net current from sodium, potassium, and the leak channels is inward, which allows more voltage-gated sodium channels to open and depolarize the membrane up to the overshoot. This localized view of the action potential is good to understand, but in my opinion, the more interesting aspect of the phenomenon is how it propagates across the axon. Here, let's consider a longer length of the axon, which is filled with multiple voltage-gated potassium and sodium channels. When the depolarization reaches a sodium channel, it causes a local action potential to occur, which then passively propagates through the axon. The segment that just had an action potential is now in a refractory period, which is characterized by the inactivation of sodium channels and a sustained potassium efflux. During this period, the channels will not answer to any amount of depolarization until they have gone through a hyperpolarization that removes the inactivation motif from the pore. As a result, the refractory period confers directionality of the signal by preventing channels to open again and depolarize the axon in the other direction. Thus, depolarizations only impact and open the channels that are in front that haven't been inactivated. We will shortly see how the refractory period affects signaling between neurons. Now, the passive propagation of the positive charges that have entered the cell as a consequence of voltage-gated sodium channels opening depolarizes the next local subregion of the axon such that an action potential fires there as well. This continuous cycle of depolarization and hyperpolarization is passed through the axon up until it reaches the axon terminal. Graphically speaking, this self-regenerative signal carries a constant magnitude throughout its propagation, and for that reason, the signal is often said to be all or nothing. When we consider two neurons communicating with each other, the refractory period caused by sodium inactivation and potassium efflux affect how the signal is carried through the body. In this simple two-neuron system, let's consider the response from the second neuron after the first signals to it. We will assume that at rest, when the first neuron signals to the second, it is sufficient to trigger an action potential. When that occurs, we can show on our plot that this injection of current produces an action potential that travels across the second neuron. If the first neuron fires an action potential very shortly after the first one, it is possible that the second neuron might not even be able to fire an action potential. This is caused by the fact that all of the voltage-gated sodium channels in the second neuron are inactivated. This state is referred to as the absolute refractory period. In another scenario, if the second action potential is fired a bit later relative to the absolute refractory period, some of the voltage-gated sodium channels will have activated again and will contribute to firing an action potential, but because not as many sodium channels can participate, the magnitude will probably be lesser. This state is known as the relative refractory period. Finally, if the second signal is fired sufficiently beyond the refractory period, the recorded response in the second neuron will be the expected all or none response. This aspect of neuronal signaling is very important because the information in the brain is interpreted in great part through the frequency of action potentials that a neuron receives and sends. And since the refractory period greatly influences that frequency, it is very important to neuronal communication. Now, one point about the action potential process that I want to emphasize is its passive element. After the current from the voltage-gated sodium channels enters the axon, it is the passive flow of current that travels to open the adjacent channels. This passive propagation is very important to take into account because it is essentially the rate-limiting factor in how fast the action potentials travel. The velocity at which the action potential travels is more often referred to as the conduction velocity. Over time, evolution has provided two main mechanisms for neurons to increase their conduction velocity. Because passive propagation is mainly what governs how fast the signal travels, we have to bring back our passive membrane properties to understand how these mechanisms operate. To get more information on these properties, you can refer to the section on the equivalent circuit model and on the cable theory to see where these equations come from. The two main mechanisms that increase the conduction velocity are linked with the diameter of the axon and the level of myelination of the axon. Starting with the axon diameter, let's consider two distinct segments of axons with the sole difference that one is larger than the other. In terms of passive membrane properties, a larger diameter will have the effect of decreasing the axial resistance and essentially increasing the space constant. If we imagine this dotted line as being the threshold to open voltage-gated sodium channels, when the most upstream channel opens, a higher value of lambda in the larger neuron will make so that the signal decays less in terms of distance, which will be able to reach and open channels at a further distance for the same amount of time. Hence, neurons with large diameters have faster conduction velocity down their axon. Now, to understand the second mechanism, we first must discuss where myelin comes from. Myelin is provided in the body by two different specialized glial cells, the Schwann cells and the oligodendrocytes. Schwann cells myelinate individual segments of the axon in the peripheral nervous system, as well as the spinal nerves and the cranial nerves 3 to 12. Oligodendrocytes myelinate multiple axons, sometimes up to 30, in the central nervous system. oligodendrocytes are also responsible for myelination of the second cranial nerve. To understand what myelin is, we can consider its formation. If we take Schwann cells, for example, and we look at the cross-section of an axon, the formation of myelin sheets begin when the Schwann cell starts wrapping around the axon. Like any other cell, the Schwann cell has a cytoplasm and a nucleus. Over time, the Schwann cell will continue wrapping around the axon such that its cytoplasm gets displaced out and only concentric layers of membrane hug the axon. This results in the separation of an inner membrane layer named the myelin sheath and the external cytoplasmic layer named the neurolemma. Two very important proteins involved in holding myelin together are the myelin basic protein and the proteolipid protein. Between each myelin segment, you will notice that there are bits of axons that are free. These free segments are known as nodes of Ranvier, and these nodes are very important because they are concentrated with voltage-gated channels. Back to our passive membrane properties, let's consider two axons of the same diameter, but one myelinated and the other not, to see how myelination impacts the conduction velocity. First, remember that as current passively propagates, there is always some loss of current that is partially due to ionic leaks. In myelinated segments, there is essentially no possibility of leakage, and as a result, the current is better maintained. To put it in passive membrane terms, the reduction of channels caused by myelination increases the membrane resistance. Another consequence of myelin comes from the additional thickness it provides to the membrane, which considerably decreases the capacitance by separating the charge at a longer distance and reducing the electric field between the opposing charges. The impact myelin has on the membrane resistance and capacitance can be further analyzed through the time and space constants. The time constant will remain somewhat unchanged because capacitance goes down and resistance goes up. When it comes to the space constant, here again, the increased membrane resistance will yield a higher constant, and as you expect, the conduction velocity will be increased. The larger space constant in this scenario will produce the same effects as having a larger axon diameter. For a given depolarization, the propagation reaches further and allows to open channels at a longer distance relative to the initial injection. You can see in the schematic why nodes of Ranvier are so important. Without these nodes, the current would simply end up decaying because it wouldn't have been regenerated by voltage-gated channels, but because these nodes are concentrated in voltage-gated channels, the action potential can be maintained through these checkpoints. The movement from node to node is named saltatory conduction, and it is considerably, sometimes up to 100 times faster than the continuous action potential propagation in unmyelinated neurons. As a final comment about the action potential and this section on the propagation of the signal, I want to show you that by using the equations that I've presented in our discussion, it is possible to simulate through computer programs the Hodgkin-Huxley model to follow the evolution of a train of action potentials over time resulting from a current injection. One very cohesive program that I want to present to you is this Python code which neatly summarizes and illustrates very well what I have explained over the course of our discussion. In the description, I've included a link for where this code was posted originally. I've made a few adjustments on my own, but essentially it is the same code. Since this code directly models the Hodgkin-Huxley model, let's use our equivalent circuit model to illustrate the different components of the code. In Python, the code requires these following modules: numpy, matplotlib, and odint from scipy.integrate. Alright, first, we need to establish some constants in our system. The constants in our system are the capacitance, the maximal open conductance for sodium and potassium, and the equilibrium potentials for potassium, sodium, and the leak channels. The values are shown both in the code and on the circuit. Then, we can establish the equations for the transition rates of each gating variable. In this code, they have decided to use different settings in each equation in comparison to the ones I've presented to you, but it doesn't matter too much because the results are still faithful to the ones of Hodgkin-Huxley. Afterwards, we can establish the equations for the sodium, potassium, and leak currents. The sodium and potassium equations use the modified versions that we've derived from the probability model. Then, To get an expression for the injected current, we must first establish the range of time for which our experiment will last. In this code, we will consider an interval of 450 milliseconds that we will divide in increments of 100ths. Since we will integrate later, it is good to use a small incremental step. For the injected current, basically what is modeled here is many conditions that will give us intervals of current injection. In the code, we have 5 intervals. In the first interval from 0 to 100 milliseconds, no conditions are matched, so the injected current returns 0. As the time surpasses 100 milliseconds, the first condition will be met and a current of 10 microamperes per centimeter square will be sent. Then, after 200 milliseconds, the current will stop due to the second condition. Between the 300 and 400 millisecond interval, the same logic applies, but this time the value of the current goes up to 35 microamperes per centimeter square. Then, after 400 milliseconds, the current is turned off, which gives this general shape of the current injection as a function of time. Now, the next part of our code is to solve the differential equations of our model. The first important differential equation comes from the sum of all currents, in which if we substitute the definitions of the currents with what we've established, we can isolate the derivative of the membrane potential with respect to time. The 3 other important differential equations are the respective differential equations that describe n, m, and h. To solve these equations, we can use the ODINT module. For this video, not to derail too much into the realm of computer science, I will leave a link in the description on how this module works, but essentially it performs a loop over the time interval we have, and for each small increment, it computes a value at that point. Then, we can collect the values computed by the module into the variables lowercase ina, lowercase ik, and lowercase il. Finally, we can make plots of the membrane potential, sodium, potassium, and leak currents, the gating variables, and the ejected current as a function of time, using the commands of matplotlib as shown here. The generated figure of this program is the following and illustrates very well the process of signal propagation, which is the core component that we are studying in our conversation. Alright, now that we understand how the signal travels through the neuron, we need to understand what happens when the signal reaches the axon terminal. I mentioned in the very first overview that neurons communicate at synapses, which are the sites of signal transmission. I've also mentioned that the cell sending the signal is referred to the presynaptic cell, and the cell receiving the signal is referred to the postsynaptic cell. Also, recall that the space between the 2 neurons is called the synaptic cleft. There are 2 types of synapses that can be found in the body: chemical and electrical synapses. All the synapses that I've shown for the moment were chemical. To see how these 2 types of synapses differ from one another, let's first go over electrical synapses. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.